The outer mitochondrial membrane, which encloses the entire organelle, is 60 to 75 angstroms thick. It has a protein-to-phospholipid ratio similar to that of the cell membrane. It contains large numbers of integral membrane proteins called porins. A major trafficking protein is the pore-forming voltage-dependent anion channel. The VDAC is the primary transporter of nucleotides, ions and metabolites between the cytosol and the intermembrane space. It is formed as a beta barrel that spans the outer membrane, similar to that in the gram-negative bacterial membrane. Larger proteins can enter the mitochondrion if a signaling sequence at their N-terminus binds to a large multi-subunit protein called translocase in the outer membrane, which then actively moves them across the membrane. Mitochondrial pro-proteins are imported through specialized translocation complexes. The outer membrane also contains enzymes involved in such diverse activities as the elongation of fatty acids, oxidation of epinephrine, and the degradation of tryptophan. These enzymes include monoamine oxidase, rotenone-insensitive NAD cytochrome C reductase, kynurenine hydroxylase, and fatty acid CoA ligase. Disruption of the outer membrane permits proteins in the intermembrane space to leak into the cytosol, leading to cell death. The outer mitochondrial membrane can associate with the endoplasmic reticulum membrane, in a structure called MAM. This is important in the mitochondria calcium signaling and is involved in the transfer of lipids between the and mitochondria. Outside the outer membrane are small particles named subunits of Parson. The mitochondrial intermembrane space is the space between the outer membrane and the inner membrane. It is also known as perimitochondrial space. Because the outer membrane is freely permeable to small molecules, the concentrations of small molecules, such as ions and sugars, in the intermembrane space is the same as in the cytosol. However, large proteins must have a specific signaling sequence to be transported across the outer membrane, so the protein composition of this space is different from the protein composition of the cytosol. One protein that is localized to the intermembrane space in this way is cytochrome C.